Hi, this is Ben Osborne with Thermo Scientific, and in this video we will be looking at how to build a workflow for processing of SILAC data in Proteome Discoverer 1.4. So we'll start up Discoverer here, and we're just going to build a workflow from scratch. So we'll start with a new workflow, and as with every workflow, we'll have our four essential components: our files, our minimum filtering parameters, a search engine, and a false discovery rate calculator. I'll link these together. Now, in order to perform SILAC processing, we'll need two additional components. It requires an event detector, because we will be looking in the MS1 scans for our heavy and light labeled peptides, and the event detector will be doing that. And here we're going to be looking within 2 ppm. We can change this anywhere from 0.5 ppm to 4. And we require our precursor ions quantifier. There's our ions quantifier. Just pull that over here. Now, there's a lot of options for using this node, and you'll find them all here. Once we go to the Quan method box, the ellipsis will bring up our Quan method editor. Now, in this case, it is defaulted to dimethylation 3-plex, so this is where we would perform any kind of experiment where we are actually tagging the peptides and we're interested in doing the Quan at the MS1 level. In this case, we're going to be doing a very simple SILAC experiment. The most common, I think, is when the lysine is +6 and the arginine is +10. But all of the logic for this experiment can be carried over to these more complex and even less common experiments. But we'll just walk through this one here. Now, in this box, we have a lot of options. But pulling this up and choosing this pre-made method is going to give us some help in filling in the boxes. So we've got our heavy channel here. We know that we're using lys 6 and arg 10. We could modify this method here if we were using a third label that was not included in one of these generic templates that we have. We would simply add it here, but in this case we'll just go forward here. In the ratio reporting, this is whatever you decide. If you want to look at the heavy over the light or the light over heavy, you would just change the place for the numerator and the denominator. Now, under the ratio calculations, we have even more choices. We can look at the raw Quan values, which I think is always a good idea just to have those. I also like to replace the missing Quan values with minimum intensity. That way, if you don't see one of your channels, instead of getting a 0 and not getting any Quan for that, I like to have that replaced with the baseline intensity. This is very, very useful when you're looking at bacteria or cancer cells or anything with maybe that's a little genomically unstable, where there's a chance that your second cell line may not just be down-regulated in that protein a little, it may just not produce that protein anymore. Now, this is also very useful in phosphoproteomics, where some phosphostates are simply on or off. They're not, upregulated threefold. So this is a useful feature here. I typically do not use single peak Quan channels. I also do not reject all Quan values if not all Quan channels are present. You would use these if you were looking on to have a very, very strict experiment where you want to see every single peptide. In my cases, for large-scale discovery experiments, these are features I do not use. The fold change threshold for up-down regulation, this is just for your output file. If I would change this to say our fold change threshold is 1.5, anything in my report that is above 1.5 up or down regulated will be highlighted in a different color. That just makes it very obvious that you're looking at a protein that's differentially regulated, but honestly I like 2.0 for this. And your maximum allowed fold change, you can move this I think up to 5,000. I set it at 1,000, and you can also just go beyond this and set it to by saying use ratios above this maximum. Actually makes this box kind of irrelevant, so you can just check that, or you can use a fold change. Under the Quan, nearly all of these are selected, and they're very good defaults. Having our ratio counts, our variabilities. We're looking at the level of protein groups. If you know that you'll be examining proteins that are very, very similar, so you're doing a keratin study specifically, you might want to turn off the protein grouping. And here we have selected to use only the unique peptides. So we're only looking at peptides for Quan that are unique to that specific protein. I cannot think of very many cases when you would want to use all peptides, but if that does work into your experimental design, you have that option of choosing in here. Your final option box is experimental bias, and this is just how we're going to normalize our ratios. You can normalize on the median of the proteins across, or you can manually normalize everything to 1.0. This is typically what I do, particularly when I'll be processing fractions separately. I just assume that the mean is going to be at 1.0 and just normalize that. That way, when I compile all of my separate fractions together into a final report, I know they've all been normalized to 1. And really, that's all there is to it. You set your normal workflow, and and you just go. I have one example file here, and this is some QE data that has been processed. This was using the IPI database, and you see that we have our accession number, our description, score, coverage, number of proteins that this in this group, the number of unique peptides that were found. Because I had the raw Quan value selected, we get the initial area, and we get a ratio of heavy to light. Now, if I want to dig into this data a little more, this is another case where it's necessary to ungroup your peptides, because now that we have peptide and protein grouping enabled, this peptide here may actually be a composite of the number of several PSMs that can be linked to this specific peptide. So I'm going to go to the results filters, and I will disable peptide, disable protein grouping, apply, and this will let me look into the data a little bit more. Now, if we go back to our proteins, let's pick something of interest, sort them by heavy light. Let's see. Let's look at this protein transport. Now, I'm going to just pick a protein here, and a single peptide, and I can pull up a number of different features here that could not be accessed when grouping was enabled. But if we go to our spectrum used for quantification, this goes and zooms in on the MS1 level and looks at what was identified as the heavy and the light. So if we see here, this would be our light channel, and here's our heavy up above it. We can select any other proteins as well. Now you notice that, because we're looking in this tiny range of 2 ppm, we're able to find our SILAC pairs, way down in the noise. Other features here is we can get box diagrams showing our SILAC ratios for this peptide. We can also go and look at the original MS fragmentation that gave us this ID, and yeah, this looks like a pretty good ID. And gives us our Quan. So yeah, and there are plenty of other options available here under the PSM group that you can add here in the column chooser. A lot of these are hidden just so that we have a reasonable amount of data to look at, but you can build a template for the output that you're most comfortable with, that gives you the best data, and you can save that. But that's all I have on building a SILAC workflow. And as always, if you have questions, I strongly recommend that you go and look and see if anyone else has had this question and posted it to the BRIM software portal. And if you don't see it there, feel free to post your own question, and we'll try to get you an answer and help.